Hey, Dr. O here. Let's talk about protein synthesis. Protein synthesis is one of your learning outcomes. It's USLO 3.5 for this particular unit. And there's a lot of different concepts and different players that are involved. So I'd like to talk about those to help you get some clarity. When we're making a protein in the body is doing what's called gene expression. Gene expression is going to go to the DNA, which is basically a big recipe book on how to make all of our proteins. It will read the recipe, make a copy of it, and then it will make the protein. That's basically the steps involved in gene expression. Now, there are two distinct processes that are involved in making a protein. One is transcribing or transcription, and the other one is translation. We're going to look at both of these. So who is it that's making our proteins? Well, DNA is involved because it's the recipe holder, but there are also some ribonucleic molecules, some RNA molecules. So let's have a look at all of these. The first process in making a protein involves something called transcription, and we're going to need to bring up a cell and a nucleus in order to get this process started. Now within the nucleus, we're going to have our DNA. And the very first thing that we need to do is to separate the two strands of DNA. And there's an enzyme that's going to do this, and it's called helicase. Helicase has to unwind the strands, first of all, because remember, DNA shows up as a double helix formation, and the two strands are wound around each other. And then after the strands are unwound, the helicase will actually separate the section that we want to copy so that one strand is exposed. And once the section is unwound, then we can copy that section of DNA. And when we do, like I said, we're only going to copy one strand. We don't need to copy two. Now, in order to do this, we're going to need another enzyme to help us, and that's called RNA polymerase. And it's the job of RNA polymerase to call in the complementary base pair for the strand of DNA that we are copying and to put it in place and secure it. Now, we want to copy the recipe from DNA, which means that we are going to create a messenger RNA, something called mRNA. So the first question we need to ask is, how do we know where to start copying the recipe? Followed by, and where do we stop? Now, at the beginning of the recipe, there's going to be anywhere from 100 to maybe 1,000 nucleotides that are considered junk and will eventually be removed from the protein that's being made. But somewhere in those additional nucleotides, those additional bases that we see, there's going to be something that's considered a start signal. And then towards the end, we're going to see the same thing where there's some additional bases that we don't need that are not part of the recipe, and then somewhere in there's going to be embedded a stop signal. Now between the start and the stop, we're going to copy this section of DNA. And we're going to create what's called the messenger RNA or the mRNA. And messenger RNA is the recipe for making the protein that we seek. And at this point, our business in the nucleus is done. We finished the business of transcribing, and now we're on to translation, the second part of protein synthesis. And this happens outside of the nucleus, inside the cell, within actually the cytosol or the cytoplasm. Now, after we've copied the messenger RNA, we're going to bring it outside the nucleus, and we're looking for what's called a ribosome. Now, why are we looking for a ribosome? Because ribosomes contain ribosomal RNA, and ribosomal RNA is a manufacturing site for proteins. Ribosomal RNA will be able to read that messenger RNA and make the protein for us. So how does ribosomal RNA do that? Well, if it reads those nitrogenous bases at 3 at a time, it's going to know what amino acid to call in. So we could say that ribosomal RNA reads the codons to determine which amino acid is next in the sequence of amino acids. And as you can see on the slide, codons are groups of 3 nitrogenous base pairs on our messenger RNA. So now we're set, right? We've got our messenger RNA, we found our ribosomal RNA, we're reading codons. But hey, where are the amino acids? How do they enter the picture? Well, we have one more RNA molecule to talk about, and that's transfer RNA. Transfer RNA is also going to read messenger RNA and its codons. And transfer RNA is going to have a set of opposite nitrogenous bases called an anticodon, and is going to bring in the correct amino acid based upon that anticodon. So transfer RNA is going to bring in the amino acid and drop it off at the ribosomal RNA, and then leave to go find another amino acid to bring in on another day. After that codon has been fulfilled, the messenger RNA will advance within the ribosomal RNA. The next codon will be read. Another tRNA will come in and drop off the amino acid. And then the ribosomal RNA is going to join the 2 amino acids together, forming the peptide bond. And the messenger RNA will advance again. The next codon will be read, and another transfer RNA will bring in another amino acid. And this will continue until the entire messenger RNA has been read, and by the time the entire messenger RNA has been interpreted, a protein will have been produced. To review, transcription occurs in the nucleus. It not only involves DNA, but also the creation of messenger RNA. And messenger RNA is going to be our copy of the genetic code for a protein, our recipe. The next process is translation, and this occurs outside of the nucleus and within a ribosome. Here, ribosomal RNA will read the messenger RNA for the correct sequence of amino acids. Transfer RNA will also read messenger RNA so that it can bring in the correct amino acids and drop them off to the ribosomal RNA. The ribosomal RNA will create the peptide bonds between amino acids, and as each amino acid is brought in and bound together, this creates the chain that will become a protein. And with that, we are done with protein synthesis. I hope you found this little video helpful. Thanks for watching.